Good afternoon, everyone, from here in the UAE. My name is A.B. Sam Thomas, and I'm the Editor-in-Chief of Entrepreneur Middle East. It's my pleasure and privilege to welcome you all to yet another webinar that we at Entrepreneur Middle East are staging with the UAE Ministry of Health and Prevention on account of the UAE Innovation Week, which we have been celebrating just this last week here in the UAE. As you know, over the last 1 week, we have had a hackathon where we have been looking into developing ideas for the next phase of future of healthcare. We also had a webinar featuring how youth can be more incorporated in the building of these new healthcare systems and ecosystems. And today we are very pleased to have with us Dr. Eric Green, who is the director of the National Human Genome Research Institute at the US National Institutes of Health. Led by Dr. Green, the National Human Genome Research Institute is the driving force for advancing genomics research at the National Institutes of Health, which is the largest biomedical research agency in the world. We're going to be hearing a lot more about genomics and the great strides it has made in the last couple of years. But I just want to take a moment to actually introduce Dr. Green. Dr. Green has been at this particular institute for more than 25 years, during which he has had several leadership roles. He has served as the institute's scientific director for 7 years, chief of the NHGRI Genome Technology Branch for 13 years, and he was also the founding director of the NIH Intramural Sequencing Center for 12 years. So for just over 2 decades, Dr. Green has directed an independent research program that included start to finish roles in the Human Genome Project, and of course, groundbreaking work on mapping, sequencing, and characterizing mammalian genomes. Dr. Green, it's a pure pleasure and honor for us at the Entrepreneur Middle East and the UAE Ministry of Health and Prevention to have you here with us. I know it's early morning where you are. Just want to say, how are you? And welcome to this very special webinar.
Well, thank you very much. I'm fine. I'm actually a morning person, so this is it's a little early, but plenty the kind of time that I'm happy to get up and talk to people from other parts of the world, especially as we continue to exist and figure out ways to connect through these virtual environments and these virtual platforms. And Zoom certainly gives us one such ability. So I'm delighted I can share this with you. So absolutely. I hope you can now see my first slide.
Yes, we can see your first slide, Dr. Green. I just want to quickly mention to everyone attending, like we will be taking in questions at the end of this particular presentation that Dr. Green will be doing. So, please use the Q&A button at the bottom of your screens to put in any questions you have, and we'll come to them at the end of the session. Dr. Green, the stage is all yours, so feel free.
Well, thank you very much. Thank you for your kind introduction. It's a pleasure to be here. Good afternoon for those of you at your end of the world. It's certainly, I'm delighted to come share with you a lot of excitement in this field of genomics that I've been a part of, as you heard, for really over 30 decades now, and dating back to the Human Genome Project, but now even looking ahead, as I will get to at the end of my talk. All of these efforts through this metamorphosis of this field of genomics and as it's reaching into precision medicine are all about a journey to advance human health. So what I want to really spend some time telling you about today are really 3 things. I first want to introduce you historically to the first 3 decades of genomics to really set a context of where genomics has come in its relatively young age, because it is a fairly young discipline. I'm going to then spend some time talking about sort of currently what's happening. What are some of the new realities and opportunities that we are facing? And then I want to tell you about the future. And it's particularly relevant for me to say a few things about the future because our institute at the National Institutes of Health recently published a new strategic vision for the coming decade that I want to tell you a few things about. But first, let me do some historical review. Let me set a context. Let me tell you in very rapid form where genomics came from and what it has accomplished in its roughly first 3 decades as a field of study. To really go back and understand the fundamental rationale for genomics, it really comes with this idea of what is the blueprint that operates all cells in an organism. The human body has like 30 to 50 trillion cells. We know that all of them operate off the same fundamental blueprint, but they use that blueprint in different ways. But for so long, we didn't really even understand what that blueprint was. And we really wanted to get to understanding it a little better so we would know more about the fundamental aspects of human biology, and of course, use that information perhaps to better understand human disease and human health. You know, over time, we figured out that blueprint lives in the middle of the cell, and these structures called the structure called a nucleus, and it's actually carried in the cells by these smaller structure called chromosomes, which sort of are the suitcases that carry our genetic material from one cell to the next, one generation to the next. But, you know, it really wasn't a full picture in terms of what was really happening until DNA was discovered to be that blueprint molecule, the information molecule common to all living forms. And so DNA became the focus of understanding the fundamental blueprint of human biology and really all of biology. You know, big clues about DNA came in the 1950s when the famous iconic double helical structure of DNA was elucidated. It gave the last piece of the puzzle as to how it was that DNA was the molecule that could carry information about life from cell to cell, from generation to generation. And with that structural knowledge, immense amount of effort went into understand how it was that DNA actually conferred information for making cells and tissues and organisms. And in the 1960s, we figured out the genetic code, how it was that DNA encoded proteins. Proteins are the building blocks of cells and tissues and organisms. And then in the late 1980s, when I was a graduate student and a medical student, was the molecular biology revolution, where the tools for analyzing DNA, for manipulating DNA, for cloning DNA, for reading out DNA, all got more and more and more sophisticated. And in particular, our ability to actually read the DNA alphabet was key to all of this, because it actually turns out DNA is complicated, but at the same time, it's actually chemically pretty simple. It only consists of 4 different chemicals. We actually abbreviate those chemicals as A's, G's, T's, and C's. But all of DNA's complexity is in the order of these building blocks, these letters, these chemicals across large stretches of DNA. Now, as we got better and better in the 1980s in being able to manipulate DNA, increasingly read out the letters, order the letters, otherwise known as sequence the letters, we realized maybe it would be possible to one day be able to read out all the DNA of an organism. All the DNA of an organism is called its genome. And genomes can be large, but they're not infinite. And so, for example, the human genome consists of about 3 billion of these chemicals, 3 billion bases, in other words, 3 billion letters. And the idea in the late 1980s really came to the fore of maybe if we got really organized and we focused on this, we could once and for all read out, in other words, sequence all 3 billion letters in the human genome. When this idea started to come to fruition, it actually became a field. And that's when the field of genomics was born. We finally had the fundamental tools for analyzing all the DNA of an organism, that's genomics. So by 1987, this new field was launched. In fact, it was launched and named as part of a new journal called Genomics in 1987. 1987 was particularly significant for me. It's actually the year I graduated medical school and graduate school with an MD, PhD. First time genomics was used in the scientific or medical literature, actually, in this particular editorial. And the idea in the late 1980s was this new field was here, this audacious new challenge was bubbling up, read out the 3 billion letters of the human genome. And all of this got galvanized into a new international project called the Human Genome Project. This was a project that actually began in 1990. It wasn't done by any one country. Multiple countries participated in it. It was a big science, big consortium. Thousands of researchers were involved. It was very different for biomedical research, not where you had individual labs working on their own, but rather you had many, many scientists working together on a very focused goal that ultimately led to the reading out of the 3 billion letters of the human genome. Project was successful by almost any metric, actually finished earlier than anticipated. And we celebrated greatly 18 years ago when we had in hand the first sequence of the human genome produced by the Human Genome Project. So I thought it might be useful in thinking about sort of what have been the big highlights in the first 3 decades or so of genomics now that we look in the rear view mirror. And I just told you about the biggest highlight, but I want to tell you about other ones. But the biggest highlight was the Human Genome Project was successful. We sequenced for the very first time the human genome. And that will, I think, go through in history as being one of the most signature accomplishments of this field of genomics. and really represents an important milestone. But that was 18 years ago. And almost immediately after the end of the Human Genome Project, either the popular press like the New York Times here in the US or major scientific journals like Science Magazine immediately recognized one of the things that was now going to become possible. It was always envisioned that if we sequence the human genome, we might be in a better position to apply that kind of approach to the practice of medicine. So they merged together the words genomic and medicine and came up with this idea of genomic medicine. And this was sort of an idea that actually began when the Human Genome Project was being conceptualized, because the idea was too much of medicine involves managing patients, treating patients based on sort of the average patient, because that's all the information we have. But we know that each of us is a little different. We know that each of us has a slightly different genome. And could we imagine tailoring how we manage and treat patients by having some fundamental information about their blueprint, their unique genome? And that's the idea of genomic medicine. The problem is 18 years ago, despite the headlines on the covers of popular press magazines and scientific journals, the notion of genomic medicine was rather blurry. We could explain it, but we really weren't ready to implement it. But that's the state of affairs that we were at 18 years ago. But that became a very focused goal for many parts of the genomics research community, including our institute at the National Institutes of Health, focused on genomics. And the idea was, could we go through and begin to systematically put into place what would be needed to use genomic medicine, this emerging medical discipline whereby genomic information about an individual would be used as part of the routine medical care. And this generated a tremendous amount of interest, a tremendous amount of excitement, but we didn't really know how we were going to navigate that journey. We had just finished the first journey of actually getting the genome sequence, but the next journey was going to bring us towards genomic medicine. And here we had a new starting line. Now the starting line was having finished the Human Genome Project and having in hand the sequence of the human genome, having it available on the internet to anybody around the world to access and use and study. And somehow we wanted to realize genomic medicine. We wanted to put into the hands of healthcare professionals the ability to generate genomic information about individual patients and then tailor their medical care and treatment. How are we going to do that? What were going to be the steps involved? We can imagine some of them, but back then we didn't really know all of them, but we knew this was the journey that needed to be pursued. We once again realized several things about this journey, similar to the Human Genome Project. It wasn't going to happen overnight. It wasn't going to happen by one scientist or physician. It wasn't going to happen by one country. It also wasn't going to be done by one discipline. It was going to involve many, many people, not doing a sprint, but more like doing a marathon, running shoulder to shoulder over some protracted amount of time, but bringing to bear a lot of teamwork, a lot of collaboration, and eventually maybe getting us to the point where the genomic medicine could be practiced. Well, with that as the framing next phase journey for genomics, what's been accomplished in the last 18 or so years since the end of the Human Genome Project? And I just want to walk you through a few more of these highlights in very just broad strokes, if you will. The very next challenge that came after the Human Genome Project was the recognition that we weren't going to stop just having sequenced one reference sequence to the human genome. We wanted the ability to sequence individual people's genomes. But the cost of sequencing was too much. That first genome was just generated, was very expensive, but it was a big first time we've ever done it kind of a project. We sought to reduce the cost of sequencing DNA and sequencing genomes. And I'll just tell you the punchline, which is remarkable, is that over the last 18 years, we have reduced the cost of sequencing a human genome by over 1 million-fold. This did not happen by accident. We recognize that that first price tag of generating that first human genome sequence of about $1 billion, and that's about what it cost, that was money well spent, but it was not going to be the kind of test that could be used in a clinical setting. We needed to really reduce the cost of sequencing, maybe getting it down to something like $1,000, which is a cost of a reasonable clinical test like an MRI or a CAT scan or many other diagnostic tests that are done. We needed to be able to generate what we affectionately refer to as the $1,000 genome. Could we generate a human genome sequence for $1,000? Well, we knew we couldn't do it using the approaches we used in the Human Genome Project, shown here as one of the several major labs in the world that was used during the Human Genome Project that was operating 24 hours a day, 7 days a week, all year long, with hundreds and hundreds of machines generating lots and lots of data. And that was just too expensive. We needed some much different approach. And over the years, in the subsequent 18 years, we found those approaches. We found new technologies, we developed them. Some of these were developed in a very cursory way in academic labs, but more and more companies got involved and companies commercialized instruments more and more. And the rest is history, because fundamentally we can sequence a genome for less than $1,000 now. And you can see on the graph on the bottom left, the precipitous decline in the cost of sequencing a human genome over the last 10, 12 years. And it has been game changing. It has changed everything in genomics, has actually changed almost everything in biomedical research because it has become so inexpensive to generate lots of DNA sequence data. Well, with that accomplishment in mind, what it allowed us to do is to take off on this idea of understanding not just one hypothetical genome, but understanding how all of our genomes differ a little bit. Just by back of the envelope numbers, any two people are 99.9% identical at the DNA sequence level. But that also means that they therefore differ about 1 out of 1,000 bases. And across those many billions of letters, it means that each of us carries about 3 to 5 million spelling differences, places in our genome where our sequence is a little bit different. To be able to catalog those differences and be able to have information for scientists about what are the most common variants that are out there in the human genome across people across the world, we've now gone through, and the community of genomicists around the world have now sequenced hundreds of thousands, probably now we're up to millions of human genomes have been sequenced. And it has given us incredible clues about places in our genome where one person might have a T and another person might have a C and another person might have an A. And of course, many of those spelling differences really are inconsequential, but a very small subset of them actually do change the way genomes work. And some of them even are relevant for thinking about how they affect an individual's health and how they might be relevant to know when you're treating a patient for a particular disease or for a particular condition. And so we've gone from having very, very minimal understanding about how the human genome varies among people, now knowing over 100 million different places. We know where people vary and we know what the most typical variants are at those positions. And all of that data, again, is available on the internet in databases and knowledge bases. And increasingly, researchers are able to access that data and be able to figure out which of those spelling differences are functionally important and which ones of even those are medically important, and how might we use that medical information in some way to tailor the treatment of patients. But in order to get better and better at that, what we need to do is we need to better understand how the human genome actually works, and then how spelling differences in it might influence how the genome works. And here we've come a long way in 18 years. In fact, I'd say we have made profound advances in understanding how the human genome functions. But let's be clear, we still have a long way to go. It is a very complicated 3 billion letters. You know, here's a very, 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 very small representation of just a teeny part of the human genome sequence. And I can tell you, and I want to remind you, the Genome Project just yielded this, the ordering of these letters across not just 0.0001%, but across nearly all of the human genome sequence. But it didn't provide an interpretation. We knew that interpreting the functional significance of all these letters was going to take much more work than what the Human Genome Project did. Now, we've made some progress. We now know that there's about 1.5% of the letters that directly code for proteins. These are protein coding genes. We now know the human genome has about 20,000 genes. When the Genome Project ended, we had no idea necessarily how many genes there were. We've really refined that number over the last 18 years, and we know where in the genome are protein coding genes. But we've also come to appreciate that there's a whole other part of the genome that's even more complicated than genes. Sometimes people refer to it as the dark side. We know that a larger fraction of the genome is definitely functionally important. Actually, it's conserved throughout evolution, but it's functioning in ways other than directly coding for proteins. This is non-coding sequences. It's actually sequences that among its tasks include where, when, and how much different genes get turned on. It is sort of behind all the choreography why brain cells use one set of genes and muscle cells use an overlapping set of genes, and the kidney cells use yet another set of genes. And so there's a lot of complexity on how each cell uses its blueprint and uses its set of genes. It can use any of them, but it only uses a subset. That's all regulated through these non-coding functional sequences. And we're getting a better and better understanding about what those sequences are all about. And then meanwhile, we've come to appreciate there's yet a whole other second genomic code. One code is A's and G's and T's and C's in an order. But there's a whole other way that our DNA functions and can be influenced in terms of its functioning, something known as epigenomics or epigenetics. Epigenomics is when our DNA gets modified and somehow associated with proteins, and how those modifications and associations happen influence how the genome actually works. And so we're getting better and better at recognizing these decorations on our DNA. It's actually one of the ways the environment influences our DNA. The environment sometimes influences how our DNA gets modified. So more and more and more, we're understanding where are these parts in the human genome that can get epigenomic marks. And we are learning that other language that's so important in genomics. And increasingly, anybody can go to the internet, go to browsers that show and catalog information about human genome function. They're really complicated. I'm just showing you one representative screenshot. But we're starting to see an annotation, an interpretation of the human genome in very, very detailed form. And it's not that we've really figured it all out. In fact, we've only probably figured out a small fraction of it. But we're figuring out how to figure it out. And I think over the coming decade and 2 and 3, we will get an increasingly highly detailed interpretation of human genome function. And that's going to be very important for everything that we do. But the other area of highlight to really also talk about is we can superimpose information about where we vary in our genomes and where those variations happen to overlap where the genome is actually functionally significant. When does a spelling difference have functional consequences, and when does it not have any functional consequences? And we know that it's not that all variants are consequential, but a subset of them are. And a subset of those influence our traits, otherwise known as our phenotypes. It influences, what we might look like, how tall we might be, other things about our appearance, but it also influences a trait that all of us care a lot about, and that's what diseases we might be susceptible to. And so here there's been tremendous progress in the last 18 years in trying to understand which genomic variants play a role in human disease. And I can tell you we have made significant advances in unraveling the genomic basis of human disease. We certainly have a long way to go, but I do think we could certainly say we have made substantial progress. Now, to really appreciate the kind of progress that we've made, it's important to know that there's all different types of diseases out there. And one way to categorize them, although it's not perfect, but it's a nice way to sort of think about things in a very general way, is some diseases are rare and other diseases are more common. But rare diseases, it actually turns out, by and large, are relatively simple at the genomic level. Rare diseases like sickle cell anemia and cystic fibrosis and Huntington's disease, these are diseases that are rare in the population across the world. But when you really can study them at the genomic level, you realize that it's almost always due to mutations in one gene. 1 out of the 20,000 genes has been mutated, and that's pretty much what causes the disease. Yes, there might be some other variants that influence the disease. There might be some environmental contributions or lifestyle contributions, but by and large, you get a mutation in a gene, one gene, you get the disease. And here we've made tremendous progress because it's relatively straightforward now to figure out what the gene is. And to give you some flavor of how much progress we've made, when the Genome Project began, out of the suspected 7,000 to 8,000 rare diseases that exist, we knew what gene was mutated for 61 of them, 6-1, back when the Genome Project began. Today, because of a rapid acceleration of us figuring out which genes cause which rare diseases, we now know of nearly 5,000 rare diseases for which we now know the mutated gene. So we've gone from 61 to 5,000, getting us closer and closer to what we think are about 7,000 or 8,000 rare diseases that exist. And I think we will be on course to eventually figure out in every case of a rare disease, what is the gene that is mutated. But those are rare diseases, and they're certainly in aggregate very important worldwide in terms of healthcare, but even more burdensome worldwide to healthcare systems and hospitals and clinics are common diseases like hypertension and diabetes and Alzheimer's and cardiovascular disease and a whole host of mental illness conditions, as well as cancer and Alzheimer's and various types of dementia. But here it's much more complicated because it's not just one gene. In fact, it's variants in multiple genes, and it's also almost all cases a greater contribution of lifestyle, environment, and other influences. And here we've made a lot of progress, and big studies are ongoing to make more progress. But really a grand challenge for the coming decades is now being able to move from understanding simple rare diseases where one gene is involved to figuring out the complexities of more common diseases where multiple genes are involved, and what is the interplay between variants in those genes and then variants and then influences of the environment. And so what I can tell you is we have a long way to go in understanding common diseases, but we also should feel very gratified because this progression of better technologies, better understanding of human genomic variation, better understanding of human genome function, and now increasingly putting all that together to better understand the genomic basis of human disease has set us up in a circumstance where we are beginning to see examples of genomic medicine emerging. These are early examples, sort of tip of the iceberg examples, but we can now are getting to see in focus that genomic medicine can be implemented, is being implemented, and it really has made a lot of excitement about what might be possible in the coming decade. I don't have time to describe some of the earliest wins, if you will, some of the earliest implementations, but let me just give you just the general categories that we've already seen successes in. We have seen successes in genomics being applied to cancer. Cancer is a disease of the genome. We are using all these tools of genomics to better understand cancer. Increasingly, we're changing how we diagnose and how we manage patients with cancer. We are getting better and better for some medications in using genomic information to pick which medication to give a patient. There's an area called pharmacogenomics. We all know that we respond differently to certain medications, and some of that differences in response is due to things, spelling differences in our genomes. And we're starting to use, for some medications, the routine of first get genomic information about a patient, look to the key areas that might tell you which medication to give them for whatever you're treating them for, and pick the best medication that's matched to that patient because of their unique blueprint. Not hypothetical, actually happening now for some medications. Rare diseases have gone from a point where genomics really didn't play a role to now playing a very major role. Patients who are being evaluated today around the world, when they have rare conditions, can't figure out what's wrong with them, hundreds and hundreds of times a month, nowadays around the world, patients are just getting their genome sequenced. It's not particularly expensive. And in a large fraction of the time, you can actually figure out what is wrong with a patient with a rare condition simply by sequencing their genome. It's absolutely here and now. Also absolutely here and now is the use of genomics to do prenatal testing. Prenatal testing has been done for a long time, but now we're doing it in a way where we don't have to do an invasive procedure on a pregnant mom to get access to the DNA, but rather what we can do is take a little bit of DNA, take a little bit of blood, peripheral blood, just from a simple blood draw. And with these very powerful new DNA sequencing technologies, we can detect the small amount of fetal DNA that naturally sheds from the placenta and floats around in the maternal bloodstream. And that information can be used to determine whether or not that unborn baby has an abnormal number of chromosomes or has conditions like trisomy 21 and other similar chromosomal anomalies. And this is being done not just sort of occasionally, but in the world, it's expected that 5 to 6 million pregnant women will get this non-invasive prenatal genomic testing done this year. It is actually the #1 genomic medicine test because of its rapid uptake. So in short, what I can tell you about genomic medicine is there's some hot areas. That list is going to grow. We're going to see more and more and more examples of genomic medicine being implemented in the coming decade. But now I want to pivot in my talk and sort of shift gears. Up until now, I've mostly told you about the history of genomics over the last 30 plus years, and really have, I think, convinced you there's been tremendous amount of progress. If you haven't told already, you can't figure it out already. I'm very enthusiastic, I'm very positive, I'm very proud, I'm very optimistic. Now let me shift gears. I'm going to give you a few examples of some new realities. And it's not that I'm pessimistic, but I also want you to get a balanced view that not everything is simple. We have some major realities, major obstacles that we're trying to deal with. But it also brings some opportunities. So I just want to share with you some real life things we're dealing with now, because everything is not simple. First reality actually is an interesting one, is the fact that, as I've introduced to you, generating a human genome sequence, for example, on a patient is actually trivial. generating the data, reading out their letters across billions and billions of bits and pieces of human DNA in a patient. But I don't want you to think that today we can go from immediately sequencing a patient's genome to instantly knowing what it all means. In fact, we actually barely know what it means. We are just in the early days of getting good at interpreting all this. In fact, when we round on a typical patient after they have had their genome sequenced and we go and round on them in the morning, this is more like what we see in that situation. We are really not sure when we look at their millions and millions of variants that that patient has compared to everybody else, which of those is medically important. We have a long way to go to making this very routine and very simple for practicing clinicians. It can be done in highly specialized circumstances. It is not routine to clinically understand a patient's genome yet. We have major research projects, major translational research projects to try to help us figure that out, and those will be ongoing in the coming years. A second major reality, but it's also an opportunity, is the recognition that the relevance of genomics has really changed, certainly in the years I've been involved. And that's coming with some consequences. I mean, in particular, when the Genome Project began, it was just biomedical researchers that did genomics. It didn't really involve much beyond those researchers. When the Genome Project ended, we started to say genomic medicine is important. We started to bring into the field healthcare professionals to help us figure out how could genomic information be harnessed to be able to eventually implement genomic medicine. But now that we're actually implementing genomic medicine for cancer, for rare diseases, for implementing pharmacogenomics, in the case of pregnant couples, helping them with prenatal testing, all of a sudden genomics is now relevant to patients, which means it's also relevant to friends and relatives of patients. It's relevant to everybody. In fact, genomics is now relevant to society. And in particular, complexity that we're facing is that genomics is touching healthcare. And I can't speak to the nuances of healthcare in the UAE. I can speak to the nuances of healthcare in the United States, and it is complicated. And then when I learn about healthcare in other countries, I hear it is complicated. And so all of a sudden, genomics, which was once a very circumscribed scientific discipline, is now touching healthcare in systems around the world that are all complicated. And as a result, there are many issues that come up. Issues about privacy, issues about payment, issues about oversight, issues about genomic literacy, both for patients and for the healthcare professionals. And so there's all these societal things that we now are dealing with. And that is really incredibly challenging. It's a reality, but it's also an opportunity. And so there's a lot of effort in genomics now to think about what are all the societal implications, what are the ethical implications, what are the legal implications, and in the case of healthcare, how are we going to get this implemented in a complicated ecosystem like healthcare delivery in many different countries, and in some countries in many different systems, even within a given country. So it's another part of our attention for the coming decade. The third and last reality and opportunity that we really are thinking about is, I don't want to leave you with the impression that I believe that genomics accounts for everything when it comes to health and disease. I know that's not the case. The reason why I've talked a lot about genomics is what I do, but I've also had the luxury of having sort of seen a technology surge associated with this field of genomics I've been involved with that has allowed us to increasingly dissect the left side of this pie chart, which is the genomic contributions to disease. But I fully appreciate the right part of this pie chart is also important, maybe even equally important, which issues about exposures and lifestyle and social context and so forth. But here I do anticipate a technology surge. We are getting better and better at monitoring and measuring all those things and accounting for all those things. And I believe we will see a surge in our ability to do various kinds of measurements to help us quantitate the right side of this pie chart. You know, and so, for example, you need to look no further than the scientific literature to hear about all these fancy new mobile sensors and things that people can carry around, even on their iPhone, that give information about their physiology, about their lifestyle, about their exposures. And I think more and more technologies will become available. We'll be able to get much better at measuring and monitoring people's individual lives and using that coupled with genomic information to maybe come up with better ways of treating and managing patients. This has led to this notion of precision medicine. Precision medicine is sort of zooming out. Genomic medicine is just looking at precisely the genomic information about patients. But if you zoom out, precision medicine is just more accounting for individual variability across a range of different characteristics, of which genomics is only one of them. It's also variability that we have in our physiology. variability that we have in our lifestyle, how much we exercise, how well we sleep, how much we sleep, what environmental influences are affecting our lives, what about lifestyle choices like what we eat or what we do to our bodies. Imagine if we had immense amounts of data about all these things. Could we use all that information more precisely for tailoring the care of patients? Well, this has led to this idea of really researching this, doing precision medicine research and merging together genomic data, more and more technology data that we can capture about individuals' lifestyle and their physiology and their environment. Oh, and by the way, in many, many countries, we've also capturing a lot of information about people's medical care that captures a lot of phenotypic information in the form of electronic health records, which is digital information. Could we put all of these data together, analyze it using fancy, more powerful engines of computational analyses to be able to tease out things that might eventually teach us better ways of practicing medicine using what we learn about precision medicine? Well, various countries around the world are doing this. The UK Biobank is the most famous of these, where a large cohort of individuals are being studied, collecting lots of phenotypic information, genomic information, sharing that, having scientists analyze it. In the US, a number of years ago, we launched a project called the All of Us Research Project, where we're actually going to study 1 million people. We're in the midst of developing this cohort, recruiting these people. When somebody agrees to participate in this project, they agree to share their genomic data, their lifestyle information. All of this is a link to their electronic health records. And we're going to then, in a responsible way, openly share this data with researchers who agree to make sure they take care of the data and build in a lot of privacy protections and analyze it. And we believe we will learn a lot about many, many nuances of our genomics, interplaying with other aspects of our physiology, our lifestyle, and so forth. And that may empower us to be able to think of better ways, not only to understand disease, but to manage disease as well. But the UK and the US are not alone. In fact, there's a whole new international consortium we help organize of many, many countries around the world. The current members are shown here, who are doing these kinds of cohort studies to try to get them to coordinate and to share data and to do things the way we did in the Human Genome Project, that everybody's boat rises when everybody shares data and approaches for analyzing data. And so this international 100K cohorts consortium is trying to bring the world together of researchers who are doing these kinds of cohort studies. And so the big opportunity, I think, is to really think about how to do research in precision medicine that will hopefully lead one day to large-scale implementation of precision medicine, as we are seeing with a subset of this we refer to genomic medicine. So that's why I want to tell you about the present set of realities and opportunities. And let me just, in the last 5 minutes or so, maybe it'll take me 10, tell you about a forward-looking view. And that relates to sort of a new strategic vision we recently published. Let me remind you, though, that even though my story that I've just told for the last 40 minutes or so sounds very straightforward, very logical, as if everything was very linear, we knew what we were doing, we just kept building on genomics and eventually brought us to today, that's not reality, and that certainly is not research. The reality is everything I've just described for the first 30 years of genomics was not linear. It was much more like this. Lots of twists and turns, lots of things you have to adapt. We were constantly figuring out strategically how we needed to change course in order to make progress towards our goals. And in fact, strategic planning and readjusting priorities has been a fundamental part of genomics since the earliest days of planning the Human Genome Project. As we've made progress in genomics, in the Genome Project, it actually involved 3 very high-profile strategic visions that guided the Genome Project. And then when the Genome Project ended 18 years ago, our institute went through and published 2 strategic visions, one in 2003 and one in 2011, that really provided a blueprint, if you will, about the field going forward. And these visions, either for the Genome Project or more recently for our institute from these past 2 strategic visions, guided everything we did, and they represented in many cases course changes to what we had previously thought we were going to do. So we embarked on a new round of strategic planning because we wanted to start the new decade with a new strategic vision. But we did so knowing that genomics has changed as a field considerably. You know, back in the days, the Human Genome Project, very, very confined project and field of genomics. But once the Genome Project ended, the threads of genomics really started to get woven more and more broadly into all of biomedical research, brought up into basic science research, human genetics research, data science and computational biology, and then, as I've alluded to, increasingly into medicine. And really, as we now exist, it is so many different disciplines, so many different types of professionals are now weaving the threads of genomics into what they're doing in research, in medicine, and as I refer to as even in society. And so it's no longer a small field. In fact, biomedical research has genomics woven throughout all aspects of it, as does medicine, increasingly as does society. So when we started strategic planning for the next decade, we did so not saying we could do strategic planning for all of genomics, way too big. We were going to simply focus on the forefront of genomics. We were going to focus on the cutting edge aspects of genomics that our institute at NIH, which is responsible for leading human genomics research in the US, should focus on. For 2.5 years, we engaged the community, we collected a lot of information. And then in late October of last year, we published a new strategic vision. We have a website that describes this in detail. If you want a copy of this 10-page strategic vision for the future of human genomics for the coming decade, very much framed around the forefront of genomics, you can go to this URL shown on the right, and you can download a PDF and read that paper, and I strongly encourage you to do so. In that paper, we describe a number of elements, the highest priority things that need to be done in genomics for the coming decade. They come in different categories. Some of these involve guiding principles and values that undergird the field that we need to articulate and enhance and really guide the field in sort of an ethical framework, thinking about key principles and values. We have responsibility at the forefront of genomics to keeping a foundation for genomics strong, fortified, building it stronger and stronger, because everybody uses genomics now in biomedical research. We need that foundation to be as strong as possible. We also have a responsibility for identifying what are the great barriers that need to be broken down. 18 years ago, we identified the barrier was the cost of DNA sequencing, and we knocked it down to $1,000. What are the current barriers that if we could knock them down, you would see everybody be able to do more and more powerful genomic approaches and methods in their research? And similarly, what are the big compelling projects? Being at the forefront of genomics means you articulate some of those projects, like the Human Genome Project and the many projects that have followed. What are the big ones for the coming decades? And we describe some of those as well. And all these come together and push us to the forefront of genomics. And then at the very end of this strategic vision, we did something we haven't done before, but we thought it would be fun, is we actually came up with a series of bold predictions. Actually, they were really aspirational and intended to be inspirational. We knew they were high risk. We knew most of them wouldn't come through, but we wanted to show people what we were reaching for. What were some of the big, bold 10 things that if we could accomplish even a subset of them would be truly transformative? And so we listed 10 bold predictions. And so to sort of round out and close my talk, I thought I would share with you a promotional video that the Institute put together internally to promote the strategic vision, but to build it around these 10 bold predictions, which have caught a lot of attention and really have people quite excited. And you will see it's narrated by a famous author who's good friends of the Institute. So let me share with you this short 3-minute video.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future, a future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2, the role of every gene in the human genome will be known. Prediction 3, environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4, genomics will no longer use social constructs such as race in human research studies. Prediction 5, student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6, genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7, it will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8, a person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9, advances from human genomics will benefit all of society and not just a few. Prediction 10, genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So I hope you enjoyed that video. And if you're interested in even hearing more about these 10-bold predictions, our institute just recently launched a 10-part seminar series. And over the rest of this year, we will be holding in virtual form seminars. Each seminar session has 2 speakers and focuses on one of those 10-bold predictions. All these are being video recorded and uploaded on YouTube and also available through our website and our Genome TV channel of YouTube. And if you look at this URL, that's where you could find it. We've had the first of 10. The next one will be next month, and I encourage you to attend these or watch the video recording of it. And so bottom line is we believe these first previous chapters, these strategic visions were instrumental in helping the field of genomics, and we think this latest one is going to be similarly extremely helpful for the field of human genomics for the coming decade. And so let me just wrap up and conclude and say it's been delightful to be speaking to you today. I look forward to some of your questions, but if you're interested in staying connected, both with the Institute and with me, I put out a monthly newsletter that my staff helps me put together, and we email that out. If you want to subscribe to that, you'd be welcome to. We will certainly in the coming years be describing some of the ways we're implementing this new strategic vision. Similarly, if you're into social media, I will similarly be promoting lots of things in genomics and lots of things about the strategic vision implementation on Twitter, if you want to follow me on Twitter. So I will stop there, and I will look forward to answering some of the questions that you have. Thank you very much.
Thank you so much, Dr. Green. I am just going to slide. Sorry. Okay, cool. Thank you so much, Dr. Green. That was a really, really insightful presentation. I especially was taken in by the last 10-minute video that you said. And like you said, the surprise appearance by Siddhartha Mukherjee was nice. For those of you who are watching, Siddhartha Mukherjee is the author of the book called The Emperor of All Maladies. He won the Pulitzer Prize for that. And it was amazing to have his voice narrate the future that we can expect in terms of genomics. I'm going to dive in straight to the questions, Dr. Green, if that's okay. And the first of these is coming in from Dr. Hinda Dagag. I hope I pronounced your name properly, Dr. Hinda. But she's the corporate genetic project head at the Abu Dhabi Health Services company, SEHA. And one of the questions she had was like the UAE here has started like various population-based sequencing programs, such as the Emirati Genome Program. Given that the UAE is like embarking on this, significant step in their research, what are some of the lessons or some of the key factors that you would, advise them to consider while setting up these large programs? Because as one of your last screens showed, the road to the last couple of years has not been linear. So if there were any mistakes, that you would tell us to be wary of, what would those be?
Yeah, well, it's hard for me to give specifics, but let me give you a very general piece of very strong advice: is don't do anything without talking to people. I think the genomics community is so well known for being incredibly open and very interested in sharing and collaborating and really constantly trying to help everybody else out. There are at least and one of the things immediately to do is join these consortium that are trying to put together at a global level different countries and different efforts. We have one in genomic medicine implementation. There's a global genomic medicine consortium, G2MC it's called. And that's one that really focuses on genomic medicine implementation. If this is a more of a cohort-based study, look into joining this IHCC, this International 100K cohort consortium, which I talked about and showed a slide for. So and then there's other similar ones. I mean, there's major international consortium for rare diseases. There's major international consortium for understanding the function of the human genome. Is try not to do it alone, because there's, and by the way, my institute has worked with other funders around the world to establish these consortia for exactly this reason, is because we all have so much to learn from each other, and the challenges ahead are so monumental. We need all of the world's intellectual capital involved in doing this. And so please join and participate. And the one thing I would also say is, maybe the more Zoom-based comfort we've gained over the last year might help these efforts, because previously we developed these consortium and then we'd hold meetings. You'd have to hold it somewhere in the world, and it's hard for people to travel all the way across the world sometimes, so you couldn't quite get everybody you wanted in the room. More and more, I think we might be capable of connecting these people and these different projects through these virtual formats.
Thank you, Dr. Green. Another question is relating to the whole topic regarding advancing genomics at a governmental level. And the question, the name has not been specified, but what can commercial organizations do to better support countries, especially across the Middle East, in advancing genomics at a government level? I presume education would play a large part, but what else has worked for you in the United States and perhaps other countries of the world that you are aware of?
Yeah, I already sort of joked that healthcare systems are so idiosyncratically unique in each country. And of course, I'm going to tell you the obvious when I say so are governments. I mean, and so, what I would say is I think a big responsibility we have in leadership positions, whether it's government leadership, scientific positions, or academia, or also the private sector, is we have to be prepared to educate. We have to be prepared to engage. We can't just go asking for things. We need to be constantly trying to help them, decision makers, policy makers, whether that's in a healthcare system or whether that's in the government, understand what these opportunities are, and to try to dissuade some of their fears and concerns and really try to help them work through them and address them. And so I think, I think it's really important to recognize we need all those different sectors involved. You can't just listen to government scientists like me. You can't just listen to commercial scientists. You can't just listen to academics. We need to be figuring out how to work as networks to help get everybody comfortable with these things. But that everybody includes the decision makers. It includes the healthcare professionals. It also includes the patients, which means we really need society to better understand these things and get to feel more comfortable with them.
Dr. Green, I know you mentioned this in your presentation as well, but there are quite a few questions that is going into the ethical dilemma, if you want to call it, regarding genomics, and how, does this actually mean that we could potentially be, creating a superhuman of some sort, or, so on and so forth. The imagination runs wild. What is your personal take on this, or like, what's the Institute's personal take on this? Like, how are you going about, tackling these difficult questions, so to speak?
Well, I mean, the difficult questions range from how do we prevent people from doing nefarious things and creating superhumans to probably slightly more down to earth practical things of how do we ensure privacy.
Exactly.
Information. I mean, and those are two very different things. I think I think superhumans are low risk. I think privacy violations could be more realistic because we see privacy violations in our financial records and other bits of our lives. The first thing you start, and it's why I put in the reality number 2, is that we have to recognize that we have a responsibility as we are advancing genomics to not do it blindly, but to do it with a societal lens, to be thinking about the ethical, the legal, the social implications of these advances. Genomics has been good at that. Even the phrase ELSI or ELSI, ethical, legal, social implications, is a term of art. We have an ELSI research program. At our institute, we dedicate 5% of all the research we do goes to ELSI research. And we are trying to anticipate what are some of these concerns. How do we better get ahead of concerns about privacy? How do we better get ahead about concerns about genomic information being misused in various ways? How do we better understand how to get ahead of misuses of technologies like genome editing technologies, like some sort of other modifications of our DNA for things that are not for medical purposes? And having research in advance helps really make those conversations much more sophisticated, because you can have real information and real data at the time where you're trying to develop policies. And so first thing is an absolute commitment to studying these things and being prepared to address them. The other thing is that you just got to deal with them one at a time, and every one of them is complicated. We are working very hard at building in all the right privacy protections, as we are in other parts of our lives, including our medical information, even independent of our genomic information. And then you look at problems like a superhuman. That's so technologically science fiction, because we barely understand. We wouldn't really understand how to modify a person to change their height, because height is so complicated, involving hundreds of thousands of genes. So we also have to calibrate what's the likelihood of there being a problem. Now, in the case of genome editing, that is a real problem. Germline genome editing has been a problem, and there's an example of this recently. And so we've mobilized the scientific community to try to put in better protection so this doesn't happen.
Dr. Green, I'm aware that we're running really short to time, but again, I'm just trying to club a couple of the questions together. And there's again, this kind of relates to the predictions that you were saying up to 2030. And one of them was like a genomic test is going to be as standard as a blood test, and having it on your smartphone, so to speak. A bunch of questions are coming out like, is this actually going to be able to prevent a variety of maladies? Like one question has been about learning disabilities, another question about psoriasis. So how much of that can one expect? What should be one looking for when we are talking about these kind of potential future scenarios?
So the short answer is, of course, we don't know. I mean, we know for some rare conditions, and those are very motivating. And we know for certain drugs, helping us decide which drugs. I think as we learn more and more, we might see a tipping where we'll say, wait, instead of getting the genome sequence when we actually encounter a problem or when we encounter the need for medication, maybe we should have it up front. Maybe we could then anticipate. There's a lot of interest and a lot of studies going on now of saying, can we use genomic information to help us sort of better risk stratify who's more likely or less likely to get diabetes, who's more likely or less likely to be hypertensive. And instead of waiting until they have that condition, maybe if we knew about it 5 or 10 years in advance, maybe we'd be able to change some things in their lifestyle that may mitigate the severity of their disease. Now, that's all a hypothesis, so I can't predict for sure that's true, but we have empowered, especially in these large cohort studies, the ability to be able to see whether that might be possible. And if we think about what happens in genomics over a 10-year span, maybe by 2030, that might be possible. And if so, we might tip the balance and be able to say we are routinely getting genome sequencing done because we're using it for A, B, and C.
My final question, really, and this is going back to Dr. Hinda Dagag, who had asked us the first question as well, was with regard to the subject of genomic medicine and its appeal here in the UAE. The UAE's Ministry of Health and Prevention, whom we have partnered with for this movement, has been like putting innovation at the forefront. But having said that, this is still very much, I want to say, a niche field, so to speak. So again, in terms of your experience, given that you spent over 2 decades in this, is like, what are some of the directives or suggestions you would have in terms of like, bringing this body of work around genomics into the mainstream and having more conversations and I want to say research about it. Like, are there any suggestions or tips you would give a body like a governmental entity like the ministry?
I think I'd go back to the answer to the first question you asked, which is, I think what you want to do is not find yourself making, sort of, don't be in a bubble. I mean, you really want to plug into these international efforts and seeing what they're learning, seeing what they're trying to implement in genomic medicine, seeing if it's gone well or not gone well. Then you could generalize it what you think it'll work well in your country, in your health system. But I think we're going to see rapid acceleration of major studies that are going to start to give insights. And I'm not saying it's going to be simple to then say, oh, well, based on that experience in that country and that healthcare system, and immediately be plugged into ours. That won't always be the case, but maybe it needs to be adapted. I just think what's going to happen in terms of the arc of knowledge that we're going to get about where and how genomic medicine, precision medicine is capable of changing the course of medical management. You want to learn about that as early as possible and then evaluate whether that would be beneficial in your country, in your health system, and in your context.
Thank you so much, Dr. Green. It's been an absolute pleasure and privilege. I think all of us have learned so much, and of course, the interest to learn more is there. And we definitely hope to attend some of your future seminars in the next couple of months. Dr. Green, again, thank you so much for being part of this event hosted by the UAE Ministry of Health and Prevention and Entrepreneur Middle East. Any final words? And
no, I would, please, stay connected. I gave you information, stay connected to me. Also stay connected to our website. It's an easy website to remember, genome.gov. We really try to put as much materials in there and lots of pointers there, and not just for people here in the US, we do that for people around the world. Also with all the videos and all the outreach we attempt to do to really share as much information worldwide.
Thank you so much, Dr. Green. For those of us who are tuned in, we will be leaving links to the website as well as the direct Twitter link to Dr. Green on our social media profile. So it's at ENT Magazine ME. You'll find all the details there. Once again, thank you all for taking time out of your Thursdays to be a part of this event, and we look forward to staying in touch.